Hello, and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Enzyme Engineering Advancements with Xdrop and Flow Cytometry. My name is Corey Stanton, and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes, and the webinar includes an interactive Q&A session with our speaker. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to as many as possible. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. Please note that all participants are in listen-only mode and that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Samplix, who developed the content for this presentation. Samplix APS supports life sciences and medical research communities with microfluidics-based solutions for detailed insights into cells and genomes. Working with academic, corporate, and government researchers, their unique microfluidics instruments, Xdrop and Xdrop Sort, enable high-throughput screening with single-cell resolution. These instruments encapsulate living cells in stable, double emulsion droplets, serving as microenvironments for incubation and screening. Xdrop offers sensitivity and accuracy in functional population analysis, transforming cell analysis approaches. Our featured speaker today is Volkan Besirlioglu. Volkan received his Bachelor of Science in Bio and Nanotechnologies from the University of South Westphalia and his Master of Science in Molecular and Applied Biotechnology from RWTH Aachen University. At the same university, he then conducted research in the group of Professor Ulrich Schwaneberg, developing ultra-high throughput screening systems based on biomimetic compartments for protein engineering and metagenomics. Now, without any further ado, I'd like to hand the mic over to Volkan. Volkan, you may begin when ready.
Hello, everyone. Thank you, Corey, for the nice introduction. My name is Volkan, and I'm here and very happy to be here with you together with Samplix to tell you how the Xdrop and the Xdrop family can add value to enzyme screening campaigns. So short words about Samplix. We are founded in 2012. We are based in Denmark. And recently, last year, we also have an office in San Carlos in California. And basically, Samplix supports the life science and medical research community by offering microfluidic-based solutions designed to deliver the highest resolution insights into cell and genomes. At Samplix, we are a multidisciplinary team of industry experts. We focus on the development and providing tools for biological research. And most of the team is based here in Copenhagen. So Samplix offer two different platforms for droplet-based workflows. First, the Xdrop, which is a droplet generator. This instrument is very useful in order to for sample encapsulation, for example, for single cell encapsulation. And besides that, we have an Xdrop Sort. This is a droplet generator and a sorter for high throughput screening applications. The Xdrop Sort is also capable of sorting droplets based on their fluorescence readout. Positive hits can be then sorted out and further characterized like sequenced or grown, for example. Droplet generation and sorting requires appropriate cartridges and reagents, all available from Samplix. Our cartridges enable 8 different sample runs in parallel at the same time, enabling also parallelization and increased throughput. And as I mentioned, we have also, I did not mention, but we have also digital tools on our website to support sample preparation and assay design. Let's focus now on the let's address the challenge in enzyme screening. So discovery of novel and improved biocatalysts traditionally has a challenge here. So traditional methods explore just a few thousand variants, leaving the majority undiscovered. Additionally, bulk cultivation complicates finding rare and active enzymes, making it finding like a needle in a haystack. So we as Samplix are here to offer a solution for this with a groundbreaking technology that enables function-first screening of millions of variants per day, reshaping the way we approach enzyme screening. So we aim to streamline enzyme screening processes. Next, I will present you first the agenda of this talk today. First, we will explore directed evolution, which is a tool of enzyme engineering. Afterwards, we'll focus more on the droplet microfluidics that enable high throughput analysis. These droplets are then further analyzed and are compatible with flow cytometry. Afterwards, we will focus more on the screening workflow with the Xdrop, and after that, we will delve into a cellulase case study that was in collaboration with my former research group. And in the end, I will give a short conclusion. So let's start with directed evolution. So before understanding what directed evolution is, let's start first, what are enzymes? Enzymes are key proteins that are involved in different biochemical reactions in living systems. A natural enzyme sourced from cells functions seamlessly at specific ambient temperature within a defined pH range and in aqueous solution all over the globe. So contrary to that, what we need in industry is a total different condition from what we have in nature. So enzymes were not evolved to function at high levels of substrate, to the exposure of organic solvents, or to be stable and have a long shelf time, for example, in our daily products. So that's why the solution is to improve enzymes and to engineer them to perform in industrial applications, to have a greener production of fine chemicals, pharmaceuticals, and for example, in the food industry. And the solution for this one is, alongside other methods, directed evolution. So let's understand directed evolution. What is directed evolution? Before starting to explain this, direct evolution is a powerful method. It was awarded 2018 with the Nobel Prize. The pioneer of this method is Francis Arnold, and it comprises 3 steps. First, we start with a parent DNA encoding for the protein of interest. This DNA is then, in the first step of the diversity generation, randomly mutations are implemented into the genome. And this yields to a mutant library. Each mutant has different specific properties on protein level. So this DNA is then used to transform the host, and under application conditions, these libraries are screened. So the credo of directed evolution also is this: you get what you screen for. So if you want to find an enzyme that is thermostable, then your assay should reflect this condition. So after screening and finding and identifying the improved or beneficial variant, either these variants can be isolated when a satisfactory improvement is reached, or iterative cycles can be performed by starting a new round based on the mutated variant before. So the bottleneck in directed evolution is the screening. This is because the more you screen, the better the improvements, simply spoken. So in traditional directed evolution, we have major challenges. So most of the time we have low mutagenesis frequencies, leading to only few amino acid exchanges on protein level. This means the likelihood of beneficial exchanges leading to beneficial improvements are lower. So often, because of the bottleneck character of the screening, often small libraries are screened. And to reach satisfactory improvements, often 3 to 6 rounds of directed evolution have to be performed, which is really time consuming, because per round 1.5 to 2.5 in improvement is on average reached. So in order to have an industrially interesting, attractive improvement, we have to perform iterative rounds here. And this takes time, as I mentioned earlier. So, and with this limited screening capacity, often the improved variants are not really understood. The functional relationship is not really there, because we can get not much knowledge out of the screening throughput. So, and typically, all in all, the time requirement typically for direct evolution campaign, for one round of direct evolution, requires 1 to 2 years, including the screening development, the assay development. So let's explore the vast sequence space and why we need to increase the throughput in screening. So let's imagine a hypothetical protein consisting of 100 amino acids. We know that proteins are built of the building blocks of proteins are amino acids. So let's imagine on each position we exchange all the different amino acids, the 20 amino acid building blocks. This leads to an unbelievable diversity, right? An large diversity sequence space. And actually, these are more combinatorial possibilities than atoms available in the universe. So in a typical diversity generation phase, these numbers are generated, for example, in a error-prone PCR. These variants are generated, and then afterwards, due to the transformation limitation, this is the variant that we can screen. So in order to screen still this very high number of variants at Samplix, we developed droplet microfluidics that enable high throughput analysis. So droplet microfluidics is an integral part of what we do here at Samplix, and many areas of modern research also focuses on high throughput analysis and development that enables the screening of millions of individual assays that need to be performed in order to screen samples to increase the analysis depth. So to perform a high throughput screening campaign, the variants that I showed you that were generated needs to be screened. And with traditional screening formats, we can not reach really the throughput that is generated. So we cannot cover really the generated diversity. And apart from that, the reagent volumes are higher, and alongside with the costs and the time. So the screening campaign with the traditional method needs longer time. Contrary to that, droplet microfluidics allow a substantial miniaturization and parallelization of the assay. The assay and single cells can be compartmentalized into thousands of picoliter droplets per second. This increases the throughput and allowing researchers exploring larger experimental landscapes while reducing also the costs per assay. So at Samplix, we focus on the generation of double emulsion droplets. In this animation, you can see the process of double emulsion generation that involves 2 different emulsification steps. In the first emulsification step, a primary water oil solution is formed where the aqueous phase it contains the sample. And then this is then emulsified in a second on the second phase here, together with another water layer leading to the production of double emulsions. So the spray size geometry of our cartridges allow control production of the double emulsions and we can produce thousands of double emulsion per second. So here's a short movie that demonstrates cartridge preparation for droplet production in real time. For sample preparation, it requires Samplix cartridges and Samplix reagents. Each cartridge can run up to 8 different samples. So you can prepare only just one sample or all 8 at a time. Only 3 reagents are required for the cartridge preparation. Once the reagents are in, the cartridge is sealed and placed into the instrument, the Xdrop. Afterwards, the user can select the program and the lanes to be run, and after 40 minutes, up to 40 minutes of production, droplet production, the cartridge can be taken out, and the droplets are ready to be collected and to be processed further. So at Samplix, we have different cartridges for the production of different emulsions. For example, single emulsion as well as double emulsion, as I already mentioned. So in this talk, we will focus on the DE20 cartridge, which produces the smallest droplets that we have. We can produce 8 million droplets per run, and this in parallel, so 8 times 8 million droplets in 40 minutes. Basically, we use these droplets for the encapsulation of DNA or microorganism, alongside with other cartridges, which are used mainly for mammalian cells and for DNA in case of the single emulsions. So let's continue with droplet flow cytometry. Due to the monodispersity, the droplets act as microreactors, like individual wells in a microtiter plate, where single cells are encapsulated and cultured and analyzed. Moreover, in particular for double emulsions, it is important to know that they are suspended in an aqueous phase. So this makes them compatible with flow cytometer, and everything is very easy to handle. The droplets are stable and biocompatible. As I mentioned earlier, the double emulsion droplets are dispersed in an aqueous outer phase, which allows integration with flow cytometry. The advantage of this workflows is that the genotype-phenotype linkage stays retained during our workflows. So the screening workflow of our study here that I will show you in a few minutes is depicted in the scheme. So we start, as I mentioned also with the directed evolution slide, we start with a mutant library. We transform the host with the library, encapsulate the cells, culture them in the cells. Afterwards, we can perform the enzyme assay inside the droplets after culturing and growing the cells and in the protein production. And afterwards, the droplets can be opened up, the cells can be recovered, and we can continue with further characterization. Our instruments enable a very fast and straightforward way to do function first analysis. It requires only few pipetting steps into the cartridge with few reagents also, and we can produce 8 million droplets per lane. So after encapsulation, we have the option to sort the droplets either on our Xdrop sort instrument, which sorts them after fluorescence readout, or we can use, as I mentioned earlier, a standard flow cytometer, a cell sorter, basically what we did here in this study. So let's come to the cellulase case study. This case study was performed in collaboration with the Spanenberg group at RWTH Aachen. And we chose a cellulase as a model enzyme here. It is a quite interesting enzyme. Cellulase belong to the third largest industrial enzymes, and they have a significant application across different industrial processes, including food, textile industry, pharmaceutical, and biofuels, and paper production. So how we used Xdrop in our case study was after assay design and library design, we used it as a pre-screening method in order to clean up the library. So basically to clean up the library from inactive enzyme variants in order to have in the end an enriched library to continue then the screening under more defined, more closer to application conditions. This is what we'll focus on in this case study. So our model cellulase was extracted from a metagenome library. It has endoglucanase activity, meaning it cleaves randomly bonds at amorphous sites in cellulose or creating new chains. It has different domains here. CBM, it is a carbohydrate binding module. This directs insoluble cellulose polymers to the core domain. This enzyme doesn't have a catalytic triad, but a catalytic glutamic acid. And this is the main point here. The aim of the study was to identify and to enrich rare variants for next round of screening. So in order to perform droplet microfluidics and screening in droplets, several steps have to be considered here. And this is what I will show you in the next few slides. So we will go through different experiments that are an integral part of designing such experiments. So let's start first with partitioning of fluorophores. Initially, we started out by encapsulating a large polymer, in this case, FITS labeled dextran, in order to see how the droplets behave, whether they are stable and maintain their integrity over a different time span here, over 3 hours. So we saw the droplets are stable, and we proceed with the analysis. Afterwards, and this is important in terms of later, in terms of the assay that we will perform, that I will show you. So we chose different fluorescent molecules that mimic the substrate that will be used later in the enzymatic assay. So they have different chemical properties. So we started out with fluorescein, encapsulated them into the droplets, and analyzed if they leak out or if they stay inside the droplets. We saw that they are the partitioning is well. And afterwards, we continued with the analysis of resorphin, and here we saw a significant leakage after 2 hours. Still, for quick assays, it is something that I would also consider to use. But the best candidate was here fluorescein. In comparison, for example, to 4-methylumbelliferone, this directly leaked out. So after knowing which substrate we could use that stays also inside the droplets, also we know that the droplets are stable. Now we went over to sorting droplets in order to see if they are stable during flow cytometry analysis and sorting. So for this, we encapsulated resorphin and FITS in 1-to-1 ratio in droplets and analyzed them by FACS. And important is here that our droplets are also compatible with various flow cytometer instruments. So this was before the sorting. This is microscope analysis. We can see the populations of resorphin containing and fluorescein containing droplets. After sorting the fluorescein containing droplets out, we got a 4.7-fold enrichment. proving that the system is capable of sorting the droplets out that contain the molecules of interest. So afterwards, building on our methodological design approach, we transition to the next step. Here it is sorting of cells and recovery. So we now encapsulated EGFP producing E. coli cells into the droplets and analyzed them. And what we could see, they are quite happy in the droplets. They can be detected in a flow cytometer with a flow cytometer on the fluorescent axis. This is the size in the indication on the x-axis. This is the y-axis, the fluorescent signal. And after sorting out, we plated them out and just analyzed the EGFP producing cells, and we got as a proof of principle 2.4-fold enrichment. So cells are viable and they can be recovered out. This is very important for the next steps. While we seamlessly now integrate the findings from our previous experimental phases, our focus now will shift to the encapsulation of a cellulase expressing cell, and together with the fluorogenic substrate. And this substrate is basically is associated with two cellobiosis together with the fluorescent molecule that we analyzed before, that we identified as the best candidate. So the cellulase hydrolases the substrate. This leads to liberation of fluorescein that is highly fluorescent, and this can be analyzed by microscopy or flow cytometry. So we can see here on this picture a cell that was in division mode. We can identify the cells producing fluorescein, and by this verification we can move further for the library generation. So as you can remember, I told you initially, we start with diversity generation in direct evolution. The library was generated. It was used for transformation of the host. So this leads to a microbial cell library. And afterwards, we wanted to see which mutational load is the best, because we wanted to have a high mutational load. As I said earlier, most of the time in direct evolution, the mutational load is low, leading to less likelihood of great improvements. So we started already with a high mutational load, and we decided to go with 10.7 of active fraction. This means that we have 10.7% of cells that express the cellulase, and this we can see on this indicator agar plates. The active cells, the active colonies, form a halo on the agar plate, which is mixed with a substrate. So the library size was 1.5 million transformants, and with this we continued. So this library was served as a glycerol stock then, as a stock, and afterwards we went into cultivation and enzyme production in flask. We encapsulated the cells afterwards, sorted them out as I showed you earlier in principle. And what we found out is surprisingly that the active fraction decreased suddenly, because the initial active fraction was 10.7. So we found out that cells producing the active enzymes were outcompeted by inactive variants in this case. So by some optimizations we performed, we then moved on with another experiment. Here we directly encapsulated cells from the glycerol stock into the droplets using the Xdrop together with the induction agent and the substrate. Afterwards, we cultivated the cells and initiated the enzyme production in the droplets. So we just incubated the droplets at 16 degrees, and we kept them in movement. And afterwards, we can see here that cells were able to grow and build a strong fluorescent signal. And in this droplet, for example, you can see a non-fluorescent droplet, a non-fluorescent event. This means we have no activity, enzymatic activity. Afterwards, we sorted this library. So before on the flow cytometer, it looked like this. This is also a reflection of the microscopic analysis. You can also confirm that we have only rare events here. We analyzed 6 million droplets, and after sorting the 0.8% of the events out and reanalyzing them with the flow cytometer, we found quite good enrichment, I would say, up to 57.5-fold enrichment. And we could see that the sorted droplets now contain only the sorted the droplets of interest. And by this, we could clean up the library from all the droplets that contain inactive enzyme. So afterwards, we performed a functional test on the ASO-CMC indicator agar plates. So this is how the agar plates looked before. We have some halo formation, which is the indication for the activity, as I mentioned earlier. And after enrichment, we could demonstrate in this proof of principle study that we can achieve 4.3-fold enrichment of cells producing active enzyme. And with this active libraries, with active variants, we can now move on to the next level of screening. By this, we can reduce the costs and accelerate screening campaigns. And because we used a high mutational load, we can also increase the likelihood of finding improved variants. Coming to the last part, I would give you some conclusions with this last slide. So Xdrop, you could see that Xdrop encapsulates cells and assay components in the stable double emulsion droplets, up to 8 samples simultaneously. And what we also could see that we could ensure no out competition in the droplets. So we could prevent interaction also between cells. And also what we could demonstrate is that Xdrop is suitable for the screening of large libraries, isolating rare and highly active enzyme variants. And what is also a very crucial point is that Xdrop simplifies microfluidic handling. We don't have any fabrication of microfluidic chips, etc. So we can directly go into the research mode and accelerate screening campaigns. This was my presentation. I'm thanking you for listening. And I would also like to add that we have a brochure and the app note available in the webinar platform. And for more information, please contact us or visit our webpage at samplix.com. Thank you, Corey. I'm finished here.
That's great. Excellent. Thank you very much for that insightful presentation, Volkan. We are now ready to begin the Q&A portion of the event. And as we do so, I invite the audience to continue sending their questions or comments using the questions window chat box. It does look like we've already received some audience questions, so I will go ahead and start with the first. This audience member, Volkan, is asking, can you screen yeast cells as well with the Xdrop?
Yes. We can screen also yeast cells. We can screen also different mammalian cells in different droplets. We have different cartridges for this purpose. And by parallelization, we can improve the throughput too.
Interesting, excellent, thank you. Another question here from an audience member is asking if Xdrop sort could also be used for the sorting process.
Yeah, actually, yes. Back then in this collaboration, we didn't have the Xdrop sort, but it can be also used to sort. Because we didn't have the instrument back then, we used instead the flow cytometer, the cell sorter.
Okay, great. So it sounds like Xdrop sort really helped coming into the process there and being really useful. Another audience member here is asking about specifically why is it that you use double emulsions in your process?
Double emulsions can be they can be used to so the advantage of double emulsions is that we can use them, integrate them into already existing instruments like cell sorters, and they are quite stable. We also can perform PCRs inside, and yeah.
Okay, great, thank you. It looks like we have time now just for one more question from the audience here. This person is asking, how much time do you save compared to traditional screening?
Oh, yeah. Quite a lot, I would say. If we focus on the single assay droplets that were screened, it's a lot time. I mean, in a traditional screening campaign, when we would screen all these 6 million droplets we have screened, we would need months, long time. And we did this in this study in couple experimental days, basically after library generation, yes.
We actually have some time here for a couple more questions, I think. Another question just came in asking about if you work with cancer cells as well in this procedure.
Yeah, this is also a good question. We have cancer cells are compatible with our with the larger droplets that we have, the 50 droplets, and yeah, we perform also immunoassays in these droplets. So the answer is yes.
Okay, great. And you just in the answer previously, you spoke about how much time that you can save, compared to traditional screening. But this person wanted to know is, could you sort of elaborate a little bit on how fast you can actually sort?
This is depending on the flow cytometer. So, for example, in this study, we sorted at 2,000 events per second, but with newer instruments, we can increase this even more. And with the Xdrop sort, for example, we work at 5,000 events per second.
Okay, great. Well, excellent. Thank you very much for those answers. At this point, though, we have reached the end of our Q&A session. To our audience members, if we couldn't attend to your questions, or if you have any further questions, you can get in touch with the team at Samplix using the contact info on the screen. That's Volkan's email there, and I'll have a chance to follow up with you after the webinar. I do thank everyone again for participating today. As Volkan mentioned, don't forget to download the supporting material in the handouts module. There's 2 documents there. I'm adding a link in the chat box right now where you'll be able to view the recording of this event. You can share this link with your colleagues so that they may register to view it as well. And for those of you who are interested in hearing more from Samplix, you can use the second link that I've added to the chat box to register for their next webinar entitled Droplet-Based Whole Genome Amplification, Successfully Sequencing Minute Amounts of Mycobacterium Tuberculosis DNA. And this will be taking place almost exactly a month from today on Wednesday, March 20th. You'll also be receiving a follow-up email from Xtalks that will provide access to today's recording. And as we wrap things up here, please note that a survey window will appear on your screen. Your participation in the survey is always appreciated as it helps us to improve upon our webinars. Now, please join me once again in thanking our featured speaker, Volkan, for his excellent presentation. We hope you found this webinar informative, and we wish you all a great rest of your day. Take care, everyone.
Bye.